I'm Jean Meserve, and I am pleased to be joined this morning by Dr. Hasham Abdallah, who is a Vice President and Head of Oncology R&D at GSK. We're going to talk about drug discovery and development. Great to have you here with us this morning.
Jean, it's a pleasure to be with you as well, too.
So cancer is sometimes described not as a single disease, but a collection of many different diseases. And even within one type of cancer, tumors can really vary in their behavior and biology from patient to patient. Does this make cancer uniquely difficult to treat?
You know, it's a really interesting question. And before I start to respond to it, I just wanted to kind of reflect a little bit on what Congressman Fitzpatrick and Congresswoman Dingell had highlighted as well, too, the personal experience. And for me, it's something I can very much relate to. I'm someone who's also lost, certainly a family member to cancer. And my father actually had colon cancer as well, too, and passed away about 15 years ago. But for me, it really brings about a really unique angle in the context of how I think about oncology research and development. I've had the privilege of being a caregiver, of being a treating physician, but also, of course, now a researcher as well, too. And so when I think about, why is it so difficult, to treat cancer? Well, part of the challenge is it's not just a singular disease. It's actually a heterogeneous set of diseases. There are actually more than 200 types of cancer that exist. And each of them have their own unique genetic profile, protein expression, methods of growth, spread, response to treatment, but also resistance. And I think that's one of the challenges that we actually tend to face as well, too. But with that in mind, I think one of the things that we certainly try to look into is how the cancer and its genetic makeup, protein profile, but also the immune system of the patient interact together. And that certainly tends to give us some additional unique insights into how we could think about treating each patient differently. Now, one thing to bear in mind, cancer cells are smart. They're highly dynamic, and they tend to evolve over time. And so what ends up happening is, oftentimes when we treat cancer with one standard therapy, these cells react to this type of pressure being applied in different ways. They may initially respond to it, but they also find ways to escape and over time develop resistance, which may ultimately either lead to a lack of response or recurrence of the disease. And so oftentimes we're trying to think about treating cancer with different modalities, different combinations of therapy that can actually apply pressure to the cancer cell through different axes, and then in turn help us make sure that patients either develop a better response, stay in response longer, or even delay the emergence of resistance over time as well.
So this complexity must have a big influence on how you as a scientist approach the whole research and development landscape. Talk to us about that.
Yeah, I mean, Jean, this is one of the challenges that we face. I think, if anything, when we think about cancer, it's a multi-dimensional lens that we have to use. It is never a single variant. It is never a single variable. And, I think probably I feel a lot of, gratitude to the fact that we live in an age where we're seeing a lot of unique insights emerge from the power of data, the power of technology. And over the past, I would say, 15 years, we've made a ton of great progress in how we're advancing precision medicine in oncology drug development. I talked a little bit about knowledge that we're gaining around the unique genetic profile of different types of cancer, the protein expression as well too. But think about this just as an example. If you think about lung cancer, it's a disease that, just to give you a sense of the complexity, it's a disease that is not only classified by the type of histology of cancer cells, but also by genetic alterations and mutations, by different types of proteins that are selectively expressed on certain cancer cells, and at the end of the day, also by the types of treatments that we use as well, too, and stages of the disease. And so each patient could be uniquely different, right? And so for us to be able to make sure that we provide the right treatment to the right patient at the right time really involves us taking a very methodical, but like I said, a multi-dimensional lens of how we think about precision medicine. And that typically involves us thinking about developing different biomarkers that can help us best elicit which patients are more likely to respond to certain types of therapies, which ones are not, which ones may experience certain side effects, others may not as well too, and then we can tailor the best treatment for each patient as well too.
You have a whole host of new technologies that have come into play. Talk to us, if you would, about how these technologies are optimizing discovery and development of new drugs.
Yeah, I mean, I would say, first and foremost, data right now in any field, that exists is a really important knowledge generator. And I think probably if you think about healthcare data, currently it makes up about maybe more than 30% of the world's data, right? So imagine the power of that and the ability to be able to tap into it as well too, and the unique insights that it could provide. Part of the challenge, that we've faced in the past is that more traditional analytic methods wouldn't necessarily be able to sort through all of this type of data. And I'll give you an example. So if we're thinking about the type of data that exists for an individual patient, cancer patient, it could be everything from what their baseline disease looks like, certain characteristics that they have, their clinical outcome, how they responded to treatment or not, what their side effects that they experienced look like, what the results of their scans look like, their laboratory values, blood tests. We talked a little bit about genetic alterations, characteristics of the tumor itself, scans that they've conducted. And so imagine trying to pull all of that data, but multiply it by the large number of patients that exist across different parts of the world, and then the unique insight that you can gain from that as well too. And so we're currently really leveraging in our field the power of artificial intelligence and machine learning to really provide us with the right, rigor and ability to detect certain patterns, trends, and potentially predictive markers of response that could then give us unique insights into what these large data sets could bring. And again, at the end of the day, one of the challenges that we have is, and this is a statistic, of course, is that as we think about the journey from discovery to approval, we're still in a stage where, any investigational medicine that enters phase 1 clinical trials only has about a 5 to 10% chance of actually making it ultimately to regulatory approval. So how do we continue to increase that probability of success? And the best way to do that is to be able to leverage this data. What's unique is we're also using this very interesting, I would say, learning iterative model, right? And it's called forward translation and reverse translation. What do we mean by that, right? Well, oftentimes we're starting first to learn about biology, again, using some of this, what we call multimodal data that's coming in from different sources, and building a computational model to help us understand disease, and then generating new hypotheses. Once we've generated these new hypotheses, we then test them clinically in clinical trials. And the data that's generated from the clinical trials goes back and feeds these multimodal data sets and feeds these computational models that we've developed to either give us new insights on discoveries and new targets that we can develop new medicines towards, give us a better understanding of how the tumor behaves and how it spreads, grows, or develops resistance, or helps us to identify why certain patients are responding versus others are not as well too.
So are these technologies allowing you to understand how different therapies will work in different patient settings?
They absolutely are. And I would say, from our perspective, we're really trying to take a very methodical approach, like I said. One of the types of technologies that we're actually using, it's very interesting because sometimes one of the challenges that we've had in this field is being able to truly replicate what happens within a patient's body, within the tumor itself, right? But doing that outside of the patient, right? And so what we've done is we actually gone through and created what we call digital twins, What do we mean by digital twins, right? Well, we go ahead, we take a specimen, a tumor tissue, a biopsy, right, from the tumor itself, take it outside, culture it through some very unique mechanisms, right, start to grow it, and it becomes almost like a living tumor that mirrors what's happening within the patient, right? And then what we can do is basically start to visualize it in 3D fashion to see what's happening inside that tumor at a single cell level, to see how each individual cell within the tumor interacts with each other. And we basically follow the patient and then follow this tumor that's outside of the patient and see how the tumor responds to external treatment and then how the patient responds to external treatment. And we gain confidence by seeing the two behave the same way. And then now this tumor that's been grown outside of the human body becomes a much more highly confident way for us to be able to test different hypotheses so that we can then take the most refined and best to then be able to apply into the patient in terms of treatment as well too.
What are some of the emerging classes of therapies that are showing the most promise?
Yeah, we've got a number of different therapies and modalities that are currently emerging as well too. And I think the fact that we've now gone from in the past and historically cancer treatment being predominantly based on surgery, radiotherapy, and chemotherapy being the mainstay of what treatment has been. We've evolved from that over the past 15 to 20 years. Our knowledge of the immune system, our knowledge of human genetics, functional genomics, transcriptomics, and proteomics as well too, has led us now to start developing new types of medicines. First, what we call a class of antibody-drug conjugates. What are antibody-drug conjugates, or as we call them, ADCs? Well, they're a really precise way of delivering chemotherapy. One of the challenges that we've had in the past with chemotherapy was it didn't necessarily distinguish between cancer cells and healthy cells. And so oftentimes we saw a very broad side effect profile from the toxicities that patients were experiencing. And so now we're able to develop these new therapies whereby we've got antibodies that are guided directly towards cancer cells that then carry the chemotherapy to deliver it precisely to the tumor cell as well too. Another class as well too, and this is an area I think where we're making a lot of great progress, is a class of molecules called T-cell engagers. These are immune cell engagers. What do they do? Well, they basically engage T cells, which are one type of immune cells in the body, and basically bring them closer together with the cancer cell by targeting a protein that is uniquely expressed on cancer cells only. And so bringing the immune cell very close to the cancer cell to make sure that the immune system is engaging the cancer cell and then killing it as well too. And what we've seen is, this class of agents is continuing to build on some of the success that we've seen recently in oncology with cell therapies, but also a class of agents called checkpoints as well too. But we believe it'll continue to at least build on the duration of benefit that patients are deriving, and again, will be applicable across both liquid malignancies or blood cancers, but also solid tumors as well. And then finally, a next generation of small molecules. And these are, at least new technologies that are selectively targeting either proteins that are expressed uniquely or ubiquitously on cancer cells and degrading them, and then thereby killing the tumor cell, or basically trying to bind this unique protein to another protein that is very essential for the cancer cell to survive and to grow, and bringing the two together. And when you do that, you're basically silencing the essential protein and resulting in cell death. So some very interesting, I would say, modalities that are emerging that I think will continue to evolve at least the treatment paradigm, and certainly have a meaningful impact on patients moving forward as well too.
So you've talked specifically about what your company is doing. Is there a need for a broader collaboration in order to really make personalized medicine more widespread?
I first and foremost, I would say innovation never happens in isolation. It can't. I think we've gone beyond a place where innovation happens in a lab within 4 walls in a singular institute. It can't. Given the extent of data, knowledge, and insight and expertise that exists across the globe. And so collaboration is a must. It's essential. It's the only way that we get ahead. And to be honest, as I think about the work that that we're doing, it's really the ability to be able to work across different sectors. So whether it be the academic sector with scientists and researchers that bring very unique expertise and capabilities, technology companies, right? Regulators, right? And of course, within even the biopharma sector as well, too. Our ability to be able to collaborate, I think, is the only way for us to be able to harness all of the power of the data, the knowledge, and the insights that exist within this ecosystem, again, for the benefit of patients and for us to be able to continue to drive new innovation in cancer as well.
Bottom line, how are these developments going to change the patient experience and improve outcomes, do you think?
Yeah, I mean, for me, I kind of I reflect on my experience in this space. I've been in oncology research and development for more than 20 years. And when I started maybe sometime in the early 2000s, we were just starting to scratch the surface of precision oncology. And I think we've made a lot of great progress since then. And it really comes down to 2 things. One, how do we really make sure that we're continuing to characterize what the patient journey looks like, right? And it's listening to that patient voice in terms of what are the experiences of patients across different stages of the disease. How are they, going through that journey in terms of the treatments, the side effects that they're experiencing, and the areas that they still feel like need to be addressed, right? And 3, how do we pull that through and integrate it into our discovery, research, and development efforts? And then the second is really about how do we make sure that we're developing the most tailored treatment plan for patients? We heard, of course, Congressman Fitzpatrick talk a little bit about the concept of screening and early diagnosis. It is extremely fundamental that we really make sure that we focus on that. It's screening, early diagnosis, early intervention, ultimately alters long-term outcomes. And our ability then to be able to tailor the right treatment plan for each patient at each stage of the disease becomes very critical. And I think, for me, the analogy is oftentimes you could think of your favorite sports team, right? Whether it be in basketball, whether it be in football, whether it be in soccer. And there's always a game plan, right? And that game plan really involves a number of different stakeholders. And in this instance, it's usually a multidisciplinary team, that is managing the patient. And being able to inform that game plan and what to do when. Are you playing offense, right? Do you have to play a little bit defense? How does it change over time, right? Is very critical. And it has to be tailored for each individual patient. And I think we're at a stage right now where we're able to do that and continue to help drive progress and innovation in the space of cancer drug development.
Sounds like a very exciting place to be at the moment, with a lot of possibilities. Great. Dr. Abdullah, thank you so much for your insights today. Appreciate it. Obviously, science, technology, collaboration all coming together with the potential to really redefine care ahead. Thanks so much. Appreciate it.
Thank you very much.
And we will now hand it back to the Washington Post newsroom.